Hello, my name is Laura Castillo. The date is February 2nd, 2018, and today's video we're going to be going over biochemical testing and microbial identification. So we did this with two different experiments this week. We did the methyl red with the bogus Proskauer test. I'm not sure if I'm pronouncing that correctly, but you know what I mean. So that was one part of the lab. And then the second part of the lab was a catalyst testing. And so with the first part of the lab, what we did was we have some existing cultured broths of E. coli and S. epidermis. We took a few droplets of those and applied them to these two tubes here. This is the MRBP broth, and this one's labeled S. epidermis. This is also MRBP broth. This is E. coli. So once we applied the S. epidermis and the E. coli cultures to these broths, we let these incubate for a little bit. I did actually let these ones incubate for 72 hours instead of 48 because my E. coli didn't really develop much turbidity. The S. epidermis did a little bit, but I thought maybe I could let them grow still a little bit. So 72 hours incubation for these, which lead me to today actually. So once I had these done, then I took 4 test tubes. And then I divided those broths. Let me not mix these here. Okay. I divided these broths that had been incubated half and half into these 2 tubes. So it gave me a total of 4. So basically divide these into 2. That's why I got those. Okay. And with that, the first part was the methyl red part. Took a couple drops of the methyl red and applied them to 1 tube each. So one of the methyl red was applied to the E. coli, and actually here you can kind of see the result. It's all kind of just a neutral yellow color, which is really the color of the dye, it seems like. So that's the E. coli. That's just the wrong one. And then this is the S. epidermis. As you can see, it's definitely more of a redder tone, and so this one did test positive for acidity. So this one's definitely more acidic than the other. Then we moved on to the next test, which was the Bogues-Proskauer test. Again, I'm trying to pronounce that correctly. For that one, we actually had 2 other little regions here. We have Barrett's A region and Barrett's B region, which I have in here because I'm not sure if we need these again, but give them there anyway. We put, first applied a couple drops, 12 drops I think it was, of Barrett's A into 1 E. coli and 1 E. coli and 1 S. epidermis tube. Of course, the ones that we hadn't used for the methyl red, and mixed those a little bit, and then we applied the Barrett's B. When we applied the Barrett's B, I did let those sit for about 30 minutes, and the results are right here. The E. coli, you can see, is actually kind of clear. There's not really much in here. So that's the E. coli, which is I'm gonna say a negative. And then this is the S. epidermis. So it's kind of hard to tell, but it did develop sort of a pinkish on the top layer. It's kind of hard to see. I was actually expecting more of a red. Maybe it's a little bit easier if I put like a little bit of white. You could see some pink in there. And so I'm gonna assume this is positive because of the pinkish. And basically that also means that this did also test for acidity fermentation, specifically, I can't pronounce that word either, butanediol. So that's for the Bogus Proskauer test. So that was that. The next part of the lab for this week was a catalase test, and that really just involved smearing 2 microscope slides with E. coli culture and also S. epidermis culture out of the plates, not out of the broths, which I found very interesting. Then we took 1 drop of hydrogen peroxide and applied it to each. And my E. coli was the one that actually bubbled really quickly, and it bubbled for a while. It was kind of a clear bubbly, but you can tell it was very oxygenated, basically meaning that was catalase positive. The S. epidermis, it actually also did bubble, but not as much as the E. coli. It bubbled for a little bit, and then it actually turned white. So, although I'm not sure if I want to say if it was catalase positive, but it was also, I guess, partially negative because it did include some neutralizing elements in there. So it did neutralize the hydrogen peroxide. And so that is what happened for this experiment. And I look forward to the next experiment. See you soon.